Hello students. After having understood the basic nature of microorganisms, their genetics and metabolism, we will cover the last topic for the course, and that is understanding how microbes cause diseases. Such an understanding of the mechanism of pathogenicity is especially important for designing vaccines to counter their negative impact. Whenever two organisms interact in a symbiotic association, there are different possibilities that can range from both mutually benefiting each other, called as mutualism, like we have seen in the case of lichens, in the case of mycorrhiza, or even the interaction between rhizobia and legumes, to both being adversely affected, called as competition. Now, along these two extremes, there are other interactions like one benefits, the other is negatively impacted or even not affected at all. In fact, in many cases, the demarcations for the interactions like commensalism, parasitism, and even mutualism, they are actually blurred. And slight changes in the environment can actually shift the association from one to the other. This module deals with the interaction called as parasitism, wherein one organism is benefited, whereas the other is inhibited or in some cases even killed. Now, there are two interactions with a plus and minus type of relationship. This is predation and parasitism. The two, as you are aware, are different in the sense that in predation, one organism is killed and consumed by the other, while in parasitism, one thrives on the other by consuming nutrients and thereby leads to a decrease in the latter's fitness. Talking about parasitism, parasites which thrive on a host can either be ectoparasites or endoparasites depending on their site of residence. The other related term is a pathogen. Now, pathogen is a disease causing organism. In fact, pathogens are a subset of parasites. Not all parasites cause disease. Can you think of some parasites which do not cause disease? Try to find some examples. Anyway, so all pathogens are parasites, but all parasites are not pathogens. Having said that, it's important to understand what is a disease. In fact, it's difficult to define a disease. Let's start with health. WHO defines health as a state of complete physical, mental, and social well being, and not merely the absence of disease or infirmity. A disease can be defined as any alteration to health. Now, diseases could be genetic, they can be degenerative, or they could be infectious. A genetic disease is caused by an abnormality in the genetic makeup, while degenerative diseases are age related. Infectious diseases are those that are caused by organisms, and we are going to talk about the way microorganisms cause diseases. So, this is our purview, the purview for today's lecture. A pathogen, like I just said, it is an organism that can cause a disease. Pathogenicity refers to the ability of an organism to cause disease or harm the host. A pathogen could be a primary or frank pathogen. These are pathogens at all times. So, whenever there is an association between such a host and between such a pathogen and a host, it always results in a disease. While opportunistic pathogens, as the name suggests, they normally do not cause a disease, but when they case of mycorrhiza or even the interaction. Two, as you are aware, are different in the is a disease. Genetic disease is caused them to cause disease or holistic pathogens, as the name suggests, a disease. Opportunities here, they normally do not cause a disease, but when they find an opportunity, they result in a disease. Opportunities here mean, for example, a weakened immune system of host or a negative impact on the normal microflora, let's say due to the administration of antibiotics, which makes it easy for the opportunistic pathogens to function. An example of such an opportunistic pathogen is the fungus Candida albicans, which is a common component of the human skin microflora. It can be both a commensal saprophyte and a pathogen depending on various factors. The fungus is a dimorphic fungus. It's an example of a dimorphic fungus. If you remember this mode of lifestyle, we covered in our lecture on fungus. And the hosts can also be of different types. A definitive host or a final host is one in which the pathogen reaches sexual maturity or reproduces sexually. On the other hand, an intermediate host is one in which the pathogen completes some stage of its development. So it's important in the life cycle, but the stages are anything other than the sexual maturity. So if there is reproduction involved in this host, it is asexual in an intermediate host. Transfer host is that host which is not needed for completing the life cycle of the pathogen, but it serves as a means to reach the final host for sexual maturity. And a reservoir host is that host which sustains the pathogen for infection in a final host. The reservoir host may or may not manifest the disease. Even the environment like soil can serve as reservoir for the pathogen. This is just to give you an example of what is meant by different types of hosts. Sarcocystosis is a disease caused by sarcocystis involving two hosts, a definitive host and an intermediate host. Humans serve as definitive hosts for some species of sarcocystis, which is acquired by consuming undercooked meat. Here, if you notice, the gametes fuse in humans, forming oocyte, which sporulates and exits the human body for ingestion by the intermediate hosts. In a similar manner, you can easily get to know if humans are a definitive host or an intermediate one for the pathogen Plasmodium, which is a protist causing malaria. Now, this is caused by 4 species of Plasmodium, and they are transmitted to the humans by female Anopheles mosquito, like you know. You will notice that in this case, while the gametes are formed in human blood, infected human's blood, they can only fuse to form zygote in the gut of the mosquito, making humans an intermediate host for the pathogen. Now, for a disease to manifest, there are 3 important components. Besides a pathogen and a susceptible host, the environment has to be conducive for the pathogen to infect and cause the disease. These 3 make up what is called as a disease triangle. Now, the disease triangle is basically a concept that illustrates the importance of all the 3 elements. So, for example, even if a host is present, it should be a susceptible one for the pathogen to cause the disease. Talking about the pathogen, different strains of the pathogen may not result in the same degree of pathogenicity or the same degree of damage to the host. Virulence is the term used for the degree of pathogenicity. And this is determined by the presence of a range of factors called as virulence factors. These could be structural features like the flagella and pili, which aids in movement and even in some cases recognition of the host, or structural elements like capsules and slime layers, which evade the host's immune system, thereby facilitating the establishment of the pathogen in the host. Then there are also factors which are secreted, which confer pathogenesis, like toxins and enzymes. They damage the host, thereby weakening it, or maybe even help the pathogen establish itself. Siderophores, which are iron scavengers, have various roles in pathogenesis. The prime being that the pathogen secreted for iron acquisition, enabling its multiplication. Maybe in this case, I should remind you of Griffith's classical experiment wherein he used two strains of Streptococcus pneumonia with the rough one devoid of a capsule, and if you remember, it was the A virulent one, while the smooth one possessing the capsule was the virulent strain. So the presence or absence of a capsule determined virulence of the organism. This is just to show you how several virulence factors can act simultaneously to work towards enhancing the pathogen's ability to establish the damage to the host. However, it is now known that virulence is not a constant property and can vary depending on the conditions. It can be lost, it can be enhanced, and if lost, it can even be restored. So the same microorganism can exist in commensal form as well as pathogenic form. In fact, this makes it challenging to quantify virulence. A commonly used matrix is lethal dose or LD50. That is the lethal dose or number of cells of the pathogen needed to kill 50% of the host. Such a quantification is, as you can understand, it is important to be done under controlled conditions. Another similar matrix is ID50 or infectious dose 50, that stands for infectious, a dose that infects 50% of experimentally inoculated cells or individuals. Now, measuring virulence, whether it is by LD50 or ID50, it's mostly done in a comparative manner under the same set of conditions. For example, which one do you think in this case is the more pathogenic or virulent one? The one with higher ID50 or the one with lower ID50? Think about it. The differences between a virulent pathogenic bacterium and its avirulent counterpart result due to differences in a small number of genes. And the genes that contribute to the ability of an organism to cause disease are called as virulence genes. Generally, the genes conferring virulence are clustered together either in groups on the bacterial chromosome called as pathogenicity islands, or they are present on extrachromosomal plasmids called as virulence plasmids. Now you can understand that by means like transformation or transduction, these genes can even be transferred into a previously avirulent bacterium. Now here there are three organisms which are closely related to each other. E. coli you are aware of. Shigella flexneri is the causal agent for dysentery, while Salmonella enterica causes food poisoning. In fact, the two are so closely related that if they are renamed as per the current systematics, which involves molecular similarity, they could be classified under the same genus. The non-pathogenic E. coli has a single circular chromosome. And if you compare the chromosome of E. coli and Shigella, there are only at few loci that differences exist. Most of the genes required for pathogenesis of Shigella are carried on an extrachromosomal virulence plasmid. While in the other pathogen, there are two large pathogenicity islands that are not found in E. coli. As I mentioned earlier, it is these pathogenicity islands which contain virulence genes. So this is how the genetic makeup would change between a pathogenic strain and a non-pathogenic or avirulent strain. Basically, the outcome of interaction between a host and a pathogen is dependent on these three factors. The number of microorganisms infecting, well, larger the number, higher are the chances of establishment and infection. However, it also depends on the virulence of the pathogen. So lesser number of highly virulent microorganisms might result into similar damage as that by higher number of less virulent organisms. And also, it's important how the host's immune system is, the status of the host defense or the ability to resist the infection. Mathematically, infection can be represented as number of organisms into virulence upon host resistance. Here is a typical disease cycle for the events driven by the pathogen. This is more or less followed by all kinds of pathogen, whether they are bacterial, viral, fungal, or even protozoans. The portal of entry of the pathogen into the host could be anything from skin, the respiratory tract, even the gastrointestinal tract, or any other opening, especially in the case of animals and higher animals. Um, Normally, the skin serves as a physical barrier to pathogens. However, in case of a wound or an injury or any other compromised situation, it serves as a conducive entry point. And after entry, there is adherence, invasion, infection, resulting damage, post which the pathogen disseminates and exits the host. Talking about adhesion, several structures like the ones mentioned earlier, for example, the fimbriae or the pili or different layers of the pathogens and appendages, they help in adhering to the hosts. In current times, you are aware of COVID-19 virus adhering to the host cells through its spikes also. Once it has adhered, depending on the pathogen and the progression of the disease using various enzymes, for example, if it's an extracellular pathogen, it breaks the extracellular matrix or the cementing material to move to other tissues. This is called as extracellular invasion. And if it's an intracellular pathogen, it moves into the cell across the cell membrane to reside either in the cytoplasm or specialized structures in the host cell, and such an invasion is called as intracellular invasion. In fact, intracellular pathogens perform both these processes, extracellular invasion as well as intracellular invasion. So here I wish to differentiate between the two terms invasion and then infection that follows. Invasion is the ability to enter, spread and cause disease, while infection is when a non-member of the local flora is established and grows in the host. Now, during invasion, there is damage to the host, but further severity is due to the release of toxins by the pathogens. Toxicity is the ability of an organism to cause disease by toxins which adversely affect the host. The toxins can be broadly classified as the proteinaceous exotoxins or the endotoxins. I'm sure you remember that we have talked about endotoxins in one of the first lectures when we were talking about the cell wall of gram negatives. The lipid A component of the LPS serves as an endotoxin, called so because it is a part of the cell's envelope. On the other hand, an exotoxin is an extracellular protein produced by the bacteria. Exotoxins typically are more specific in terms of their effects and also the organism secreting it. They are more potent in their activity than the endotoxins. The endotoxins are more general in their role. Mostly exotoxins are secreted by gram positives, though gram negatives have also been reported to synthesize them. Endotoxins, of course, they are restricted to gram negatives only. They are part of the cell wall of gram negatives. Here is a compiled table of differences between the two. Here you can notice that although endotoxins are less specific and also less potent, they are heat stable. And I will end by presenting an overview of the different stages in a disease. Here the red line depicts the number of pathogenic microorganisms, while the blue line is for severity of the symptoms. The incubation period is the phase when the number of pathogenic organisms are insufficient to cause symptoms. As the pathogen divides, the host's immune system is also triggered. Here, symptoms start appearing. However, the symptoms are more general. They're not specifying any definite causal organism. This particular phase is called as the prodromal period. What follows is the period of illness when the symptoms are the most severe. Then there is a period of decline. So, decline in symptoms, which corresponds to the decrease in the number of pathogens. And finally, there is a period of convalescence where the body has come back to normal or normalcy, but the pathogenic organisms are still reducing in numbers. That's about microbial pathogenicity for the course. In this context, I have to reiterate something. Um, as discussed in the earlier classes, you can understand that it is the environment which has a pivotal role in determining pathogenicity. And the environment favors bacteria or microorganisms due to several reasons. They tend to have an upper hand by their high growth rate and high mutation rate compared to higher organisms. Also, bacteria are much more versatile in terms of substrate utilization and also in terms of biosynthesis. Hence, they can adapt to various niches. With that remark, I would like to now end the presentation. See you all, students.